Hello Nutri subscribers, welcome back to NutriVerse Academy. So previously we discussed about nutrigenomics and also the tools and techniques of the nutrigenomics. In that we also discussed about pathway of nutrigenomics. If you didn't watch the videos, please go and watch the videos. And yes, so in previous episode we talked about proteomics. Today we are going to discuss about genomics. Okay, so in the name itself we have genomics, in the name itself we have genes. That means we are totally discussing about genes in this today's topic. So first what is meant by this genomics? So it is a study or it is an analysis, right? So it is an analysis of nutrigenomics. It helps of an organisms to identify the complete set of DNA. Okay, and next it helps to understand how genetic variations and genetic expressions will influence nutrient metabolism and diet. Okay, so that is the genomics, that the whole set of DNA will be analyzed or examined, which helps to understand how genetic variation is influencing nutrient metabolism and how diet is affecting the genetic expressions. As we know, this as I said, it helps to identify genetic variations, right? For example we have MTHFRG. Okay, so these genetic variations, if this this genetic variation is helps or affects folate metabolism. So this genetic variation affects folate metabolism, right? So that is, it helps to identify specific genetic variations. For example, SNP, a single nucleotide polymorphism. That means for every individuals have their own metabolic nutrients. That means how this genetic affects individuals of metabolized nutrients, right? So that is the best example of this. If this genetic variation is there in our body, that means it is affecting our folate metabolism in our body. So this is the example. Another example of that also, LCT genes or LCT genetic variation generally determines lactose intolerance. How we are determining that this individual has lactose intolerance? So that you no need to intake dairy or dairy products. How we are determining? By the analysis of specific gene variation, that is LCT gene variation. If this LCT gene has a variant, then it is affecting lactose in our bodies, that such that we are determining that the person or the individual is diagnosed with lactose intolerance. So it is also, these genomics plays an important role in identifying genetic variations, and also it helps for understanding diet gene interactions, as I said. So diet gene interaction is also there, as in previous proteomics, or also I was explained about flaxseeds, right? So here I'll say the example of omega-3 fatty acids, which influences genes involved in inflammation. So these omega-3 fatty acid nutrients will helps to reduce inflammation. So how we are analyzing? Through genes. Okay, so and also we after this gene expression and gene variation will give a personalized nutrition for every individuals. Okay, and it also helps in disease prevention and management. So what does the role it does? I'll write here. Okay, I'll write here. And the entire topic of these tools and genetic variation, you can check it out in the PDF, which I'll post in the description. And also there is a community called NutriVerse Academy WhatsApp community. You can also join in there for further information, and any course details or anything will be available in that channel. Okay, you can also link into the channel, and also you can download the entire topic of the tools and nutrigenomics and also the part of the genomics topic in the description. Okay, so what the role, what does the genomic does? It helps to identify It helps to identify, I think it is not visible, I'll write here, okay. It helps to identify genetic variation, and it plays an important role in diet gene interaction, and it is also plays an important role in giving personalized plans, nutrition plans or diet plans, and it also helps in prevention and management. Disease prevention. It also helps in disease prevention. So the round figure of the genomics is simple. First, as in the name itself, we are discussing about genes, okay, and it is an analysis or is a technique which is going to included with genes, okay. It is a complete set. In this organisms complete set of DNA is involved, which help us to understand how genetic variation is influencing nutrient metabolism and how the diet is affecting our gene expression, okay. As we know that every omics technology, diet gene interaction will be there, personalized plan will be there, and disease prevention will be there. Only the unique thing is the first side heading will be changed, okay. So in that, here what is that doing? It is identifying the genetic variation. As I said, the example of LCT gene and MTHR gene, so it helps to how you are diagnosed with a certain type of nutrient intolerances or something related to nutrient variation. So that like that we are identifying the genetic variation in the first topic. So everything is similar as that of the proteomics, okay. And now we are going to discuss about applications of genetic variations, and there is also some techniques which is used in genomics for which is also used in nutrigenomics, but we are not going to discuss in depth of these genomics, okay. I'll just save the side headings of the techniques. You can watch, you can also read in the PDF which will be available in the description, okay. But it is not that much important in the exam or in the, just for awareness, okay. And techniques used in genomics for nutrigenomics is, first is DNA sequencing. That means how the genetic variation is affecting the nutrient metabolism. With the help of the DNA sequencing we can know, okay. And also microarray technology is used, that means to analyze gene expression changes in response to diet, how the gene is expressing for the changing of the diet. So through microarray technology is used, and also polymerase chain reaction is used to detect genetic variations related to nutrition, and also CRISPR Cas9 used for genetic editing studies in nutrition research. So like this it will be there. And the applications of genomics, as I said, the impact of dietary polyphenols on gene regulations we can also use, and how fiber intake influencing gut microbiota related to genes. So these are some applications which is developing nutritional guidelines based on genetic predisposition to chronic diseases is also there. So by integrating these genomics with proteomics and metabolomics, which is a foundation of precious nutrition, right? So it gives the entire genetic profile of the genomics. It is just as simple, just to remember that what is the definition of genomics and what is the role of genomics and what the applications is related to genomics. This is more than enough answer, okay. So just you relate with that, okay. So that is about genomics, and it's just a simple and short topic. It is not the vast of proteomics, okay. And the metabolomics will be the some vast, and transcriptomics will be the vast, but not as the same as genomics, okay. So next we are going to discuss about metabolomics. So now metabolomics, right? So metabolomics means metabolites, right? So these metabolics metabolomics study of metabolites. That means we are going to discuss about metabolites. So metabolites is a small molecules which is involved in the metabolism, that is within the cells or tissues or biofluids. Okay, so the role of the metabolomics is to analyze the metabolomites, which is a very small molecule that are present within the cells, tissues and biofluids. So it helps to understand how nutrient and dietary compounds are affecting metabolic pathways. We have glycolysis, lipid metabolism, oxidative stress, as I said in the proteomics, right? The same thing, the metabolic pathways will be the process in the metabolomics. So the entire metabolomics travels in the metabolic pathway. Okay, it is also helps to understand the overall health of our body. Okay, so what is the role of that metabolomics? So it identifies metabolic responses to the diet. Okay, and what does it identifying? So every individual has their own metabolized in nutrients, because we have genetic variation, right? For example, blood glucose and lipid, which is responding to the individuals consuming the same diet. Some members will say, I am taking limited food, which is also affecting me to gain weight. I am taking a huge amount of food even though I am not gaining any weight. Why? Because every individual has their own genetic variation in which they responses to the different ways of thing. So how it is responding to due to this metabolic pathways or metabolites which is present in every individuals. So that's why it will rest how your metabolism responses to your body. In the same way it will respond to your body, to your overall health of your body. So that's why it helps to identify the metabolic responses to your diet. Okay, and also it also helps to understand biomarker discovery for nutritional health. So what does this biomarker discovery for nutritional health means? So generally each and every individuals have the common type of lifestyle disorders like obesity, diabetes and CVDs, right? So these will help, these metabolomics helps to identify biomarkers through nutritional health. For example, increased branched chain amino acids which will links to the insulin resistance. How you will identify that you have insulin resistance means by knowing the increase or decrease of the BACAs, that is branched chain amino acids. That is the best example of biomarker discovery for nutritional health. And next is gut microbiome interaction. So gut microbiome interaction means it helps to analyze how our gut bacteria will metabolizing our dietary components. So it will helps to identify how our gut microbiome, that means how our gut bacteria is responding to our body. For example, fermentation of fiber produces short chain fatty acids. Okay, so that influences inflammation and metabolism. So this is the best example of gut microbiome. And next, as usually, personalized nutrition also we can give. So that is about metabolomics, and we have some applications and also we have techniques which is not very important. So I'll go to applications. So applications of metabolomics in nutrigenomics, generally it helps to monitor dietary whether it is affecting metabolisms or not. Okay, like we will say to increase our metabolism, we will give ketogenic diet, Mediterranean diet, so like that. Okay, it helps to our dietary. Okay, and next is detecting metabolic imbalance, which like hormonal imbalance also can be detected. As I earlier said, biomarker discovery like insulin resistance, that is mostly seen in type 2 diabetes, right? So next, bioactive compounds also we can study. how the metabolic affecting for example flavonoids, polyphenols. For this also we can give, and it is also optimize athletic performance through personalized nutrition based on metabolic profiles. Through entire metabolic profiles of the athletic person we can personalize their diet plan. So this is the about metabolomics, which is very simple. Generally metabolomics have a very small molecules called metabolites, which is present in the cells, tissues and biofluids. These metabolomics analyzes to understand nutrient and dietary components, whether it is affecting metabolic pathway or not. So the main role of the metabolomics is to identify metabolic responses to the diet, and it also helps in understanding or discovering the biomarkers for nutritional health, and it is also analyzes the gut microbiome interaction, and it also helps in planning a personalized nutrition dietary plans to the athletic persons of their entire metabolic profile. So very small topic, and which is also very easy to learn. So this much is enough. This much is full enough for 6 marks or for 3 marks. Okay, and next we will discuss about transcriptomics. Next is transcriptomics. Transcriptomics nothing but we are learning about RNA, mRNA transcripts, right? Under specific condition the genes will be produced, right? So that's why it helps to analyze dietary components which is influencing gene expression and metabolic pathways. That means this transcriptomics helps to understand and also to analyze how the dietary components or the nutrients is influencing the metabolic pathway and also the gene expression. And what is the role of the transcriptomics means, as we said, diet gene interaction will be there, and also nutrient response, and also nutrients responsive gene, nutrient responsive genes is also will be there. I'll explain about that. And also it helps to identify the functional foods. It helps to identify functional foods. Very important. And also personalized nutrition plans based on gene expression. Personalized nutrition based on gene expression. Personalized gene expression. Okay, so firstly definition is clear, and here the role of the transcriptomics is to interact between diet gene interaction. So it will understand as to how this diet gene interaction will be there. For example, vitamin D influences the expression of genes related to immunity and bone health. Okay, that how we are saying vitamin D is influencing this and this part and this part, or this gene and this gene, through diet gene interaction. So that is the best example. That means certain nutrients regulate gene expression through transcription factor. So that is the main important in diet gene interaction. And next is nutrient responsive genes. For example, omega-3 fatty acid influences certain genes which are involved in inflammation and lipid metabolism. Through these down-regulate or up-regulate of the specific gene. So that is why the responsive genes will be interacting with the nutrients. And next is studying the impact of functional foods. So generally transcriptomics will help to evaluate how bioactive compounds are affecting gene expression. bioactive compounds like polyphenols and flavonoids. For example, curcumin, which is present in turmeric, right? So these curcumin generally modulates genes related to oxidative stress and inflammation. So which is a functional food. So what is meant by the functional food? Functional food is nothing but how the nutrient is maintaining, that it is enriched, the food is which is enriched with many bioactive compounds or nutrients which helps to diagnose our diseases. So that is the main thing of the functional food. Okay, the best example is curcumin. And also here I said personalized nutrition based on gene expression. That means each individual responds differently to the same diet, right, due to variation in the gene expression. So some individual shows increased expression of genes related to insulin resistance, which is response to high fat diet. So that is there in the personalized nutrition. So entirely with the genetic variation and gene expression. And next is some applications. So generally it helps how diet influences the chronic diseases. Okay, and it also helps us to identify molecular targets like nutraceuticals and functional foods. And it is also helps to understand dietary patterns that affects gene expression. And also it helps to develop personalized dietary strategies based on gene expressive profiles. So that is about transcriptomics, and that is about the tools of nutrigenomics, the entire topic of the new tools of nutrigenomics. So I hope you understand. If not, please comment below, and the entire notes of the topic will be available in this link. You can download the PDF and you can read, and please join in the community channel so you can get more information there. And I will also guide if you want any help from the nutrition background. Okay, and yes, that is for today. The entire topic of proteomics, genomics, metabolomics and transcriptomics as covered. Okay, and thank you, and please do like, share and subscribe to my YouTube channel, Nutriverse Academy.